Welcome everybody, this is Daniel Blackburn and this video is part of the course SWAE3411 Environmental Soil Microbiology. Today's lecture is about nitrogen cycle. First of all, it has several isotopes. Nitrogen 15 is a stable isotope, and nitrogen 13 is a radioactive isotope that decays to carbon 13. Nitrogen was first found in 1772, described, I would say, in 1772 by Daniel Rutherford, and here is illustration of the most common molecule of nitrogen in nature, which is N2 with a triple covalent bond. It's the main form of atmospheric nitrogen. Why nitrogen is important? Nitrogen is the macronutrient with highest concentration in plants, and also its concentration in microbial biomass it's even higher. We can express this amount of nitrogen in the biomass of plants and microbes by not only by percentage but also by carbon to nitrogen ratio. How much is the relationship with how much carbon and how much nitrogen each organism has. So the carbon to nitrogen ratio for plants will be much higher than it is for microbes, because plants will have fiber material, cell wall material that the microbes will not have. Bacteria, so while plant biomass could have carbon to nitrogen ratio really high, over 30 up to 100 even. Some microbes, in general, you would say that the prokaryotes, they will have like 5 or 6 carbon to nitrogen ratio, which means that it has 5 times more carbon than it has nitrogen. But if you compare that with fungi would have 10 times more carbon than it has nitrogen. So this carbon to nitrogen ratio. Why is that important? The importance is when microbes are making more cells, growing, it means that these microbes have to make cells with the same composition. It will consume 5 times more carbon than nitrogen. And usually carbon will be more abundant on the environment, and it will deplete the nitrogen, and nitrogen will be deficient on these systems. Yeah, deficient on these systems. Yeah. So nitrogen in plant physiology, but not only plant physiology, but microbial physiology also. The main forms of nitrogen inside the cells are proteins. So amino acids, the amino in amino acids stands for the amino group, which is nitrogen. Yeah, it's so all amino acids have nitrogen, and therefore all proteins are nitrogen. And this is so that for the majority of the organisms you can express even make a direct correlation, and nitrogen can be a representation of the amount of protein in cells. And if you measure proteins, that will be also a somewhat accurate representation of the amount of nitrogen. The other form of nitrogen in cells will be DNA and RNA. So unlike the phosphorus, the nitrogen will be part of the bases. Yeah, phosphorus will be part of the backbone of the DNA, but nitrogen will be part of the bases. All bases will contain nitrogen, and therefore it's a really important function to be part of the genetic code of every cell. So these two roles are the main physiological roles that it plays. But if you think about the enzymes or proteins, so all enzymatic reactions inside the cell, all the biochemistry of the cell will be dependent on if the microbes have enough nitrogen to produce those enzymes. And yeah. In the plants, nitrogen will be also part of the chlorophyll and can be continuously reused and moved around within different plant tissues. Yeah? And within the microbes will be able to eat dead biomass and cycle the nitrogen from dead cells. And the nitrogen will be fairly mobile and fairly recyclable in these systems. Yeah, very recyclable in these systems. So the chemical forms that are available for plants and microbes are ammonium and nitrate. Yeah, ammonium and nitrate, these are the main ones which plants and microbes can uptake from the soil solution. Nevertheless, other forms can be cycled into these available forms and become fairly quickly able to be uptaken by these microbes and plants. So also this nitrate can also serve as an electron acceptor in anaerobic environments and help in the respiration process. Nitrogen is often limiting in the soil conditions and also in water conditions, meaning that it's one of the importance of the nitrogen will be that you need to add it to soil system because it's always lacking. So for the importance of nitrogen, we have that economically it's the third most costly input in crop production. It's the fertilizer we add actually in highest amount in soils, and this fertilizer use is responsible for a great deal of environmental problems and pollution. Yeah? Because not much of the nitrogen that we add, the efficiency is not high, so the majority of the nitrogen that we add to soils are lost, either to atmosphere or to waters. So it's important for fertility because it's very deficient and we need to add to soils. And it's when we add it to soils, usually that is a big problem in the pollution side. Yeah, it's causing environmental problems of eutrophication of waters. If we have to resume in a nutshell what is the nitrogen cycle, we can start by looking at the biggest pool of nitrogen, which is atmospheric nitrogen, N2. There are other forms of atmospheric nitrogen, but mostly N2. This atmospheric nitrogen can move into soil and waters by the biological nitrogen fixation through bacteria that carries nitrogenase enzymes. But nowadays, a big part of the fixation of atmospheric nitrogen is industrial. It's made by humans when we produce fertilizer. Now, when this fertilizer is added to the soil or the biological nitrogen fixation fix ammonium in the soil, this ammonium can have two different outcomes. It can be assimilated by plants and can be or microbes and can be inserted and be part of the organic matter, or it can be nitrified into nitrates and nitrates. Now, the nitrates and nitrates can be either assimilated or can be leached or can be denitrified back into N2 in the atmosphere. Now, if we look at it from the microbial point of view, these processes become a little bit more complex than this. Yeah? So the atmospheric nitrogen, if you look at this figure, before we start, I just want to highlight that the white arrows are aerobic reactions, the black arrows are anaerobic reactions, and the gray arrows are either aerobic or anaerobic. So if we see the nitrogen fixation process, the biological nitrogen fixation can be aerobic or anaerobic. Assimilation can be either aerobic or anaerobic. But nitrification is always an aerobic process. Yeah. And nitrate assimilation also. So on the anaerobic side, we have more complexity. Yeah, the usual and the most well-known part of the cycle is the denitrification process, and the other arrows here were often confounded and included on the direct denitrification process, but later on it was discovered that not all nitrate losses are by the direct denitrification, but also we have other two reactions that we're going to explain in a bit. One is Anammox, anaerobic ammonium oxidation, and the other one is DNRA, which is dissimilatory reduction of ammonium, of nitrate to ammonium. So, talking about the reservoirs, we have the atmosphere is the biggest reservoir. Second, we have the ocean and the biomass in the ocean and also in the salt. And we have the land nitrogen, which will be either in the organic matter or in the living organic matter, which is the biota. The quickest cycling ones, pools, are in the ocean and in the biomass. The atmosphere pool is not quickly cycling, and also the organic matter in the soils are slow cycling, considering that the humus is not easily decomposed by microbes. So the biggest pool of nitrogen is atmospheric nitrogen, N2. It accounts for the majority of the nitrogen in the Earth systems. In the atmosphere, it's 78% by volume or 75% by weight. And it's very, very stable. The reason it's stable is because the triple covalent bond that we have between the two nitrogen atoms, and also that the lack of unpaired electrons make it really hard for this molecule to react chemically with other compounds. So it's quite, let's say, quite stable, almost inert, and it takes a lot of energy to break that triple bond. Nevertheless, N2 is not the only type of atmospheric nitrogen. We have also nitrous oxide, which accounts for 0.025% of the atmosphere. And the reason that nitrous oxide is so important is that it has a very strong global warming potential, 298 times higher than the global warming potential of CO2. Nitrous oxide is usually emitted during the denitrification process, but also it can be generated by electrical storms and lightnings, which breaks down the N2 molecules and oxidize them into nitrous oxides. The other form of atmospheric nitrogen is ammonia. Ammonia is the equivalent of ammonium. Ammonium is the ammonia dissolved in water solutions, which takes up one hydrogen from the water. So NH3 and NH4, they are in the same equilibrium in soils. If you have a high concentration of ammonium in soils, you will be expecting that this will be being transferred to the atmosphere through volatilization and leaving behind one hydrogen in that process. Ammonia is also very important because it contributes to ambient levels of fine particles and nitrogen depositions. Yeah? The main source of atmospheric ammonia is the emissions from fertilizers, especially urea and ammonium-based fertilizers. The atmospheric deposition is not very important in the majority of the ecosystems in terms of percentage-wise. It's not very high. But in some ecosystems in which nitrogen is very deficient, it can be significant. It can be a contribution as a source of nitrogen, depending on how nitrogen deficient that environment is. A little bit of nitrogen coming from atmospheric deposition is useful. In tropic climates where you have high rainfall, you can expect some wet deposition of nitrogen, but in Oman you mostly will expect dry deposition of nitrogen with the dust. Yeah, with the dust. Anyway, not the most relevant source of nitrogen for any environment. It's just worth mentioning, but not very high. Now, industrial N fixation nowadays is one of the main forms in which atmospheric nitrogen is then transferred into soil systems. Yeah? Into soil systems. The first reaction of atmospheric nitrogen industrial fixation was the Birkeland I process in 1903, and it was not very efficient, energetically speaking. In the scenario of First War, Germany charged this a chemist, very bright chemist called Fritz Haber, winner of 2 Nobel Prizes, to find a way to fix atmospheric nitrogen. That is because they were facing this distribution problem and problems with importing salítry to make bombs. Yeah, nitrogen was one of the main ways that the Germany was making bombs, and because they were having trouble importing the salítry, now they had to come up with a way of fixing atmospheric nitrogen. And Fritz Haber came out with a good process, and Bosch was the one who adopted and optimized it for industry. So that is why it's called Haber-Bosch process, which is still used today to fix atmospheric nitrogen. This process consists basically in submitting atmospheric nitrogen to high temperature, high pressures, and in the presence of a catalyst, and that will be the way that is the N2 molecule is being break down into ammonia. Now the ammonia can be further used to produce fertilizer. If it's combined with sulfuric acid, it will come with ammonium sulfate. And if you want to produce urea, then you have to react it with CO2 and have that carbon molecule binding the amino groups. Now, besides industrial nitrogen fixation, the biological nitrogen fixation is what is the natural way in which nitrogen is moving from the atmosphere into Earth systems, into the pedosphere, and in the hydrosphere also. The microbes that are cycling nitrogen from the atmosphere into the Earth systems, these microbes are called diazotrophs. These are all the bacteria, only bacteria does this, by the way. All the bacteria that carries one enzyme called nitrogenase. Nitrogenase enzyme is the only enzyme in nature capable of breaking down the N2 triple bond. This is a highly important and not very diverse biochemical pathway that is the only way in which these systems are being able to fix atmospheric nitrogen. Without this bacteria, there will, before the industrial nitrogen fixation, it will be very hard and even more limiting the amount of nitrogen on these environments. Most of this nitrogen fixation, by the way, is symbiotic. It's fixed in the symbiotic way, which means that these bacteria are associated with other organisms, and they are providing the nitrogen while the other organisms are providing sugars, easy carbon sources, and minerals for these bacteria. With plants, the most classical relationship is the rhizobium bacteria, which form nodules on the roots of legumes. So the legumes will be able to obtain atmospheric nitrogen thanks to having this symbiotic association with rhizobium on its roots. So in the nodules on the roots of the legumes, this atmospheric nitrogen will be fixed. At the same time, the plant will be providing the microbes with free sugars and minerals. But rhizobium is they're not the only bacteria doing this. We have other symbiotic and non-symbiotic organisms doing this. So cyanobacteria is also older than the rhizobium. It's probably the first microbes to fix atmospheric nitrogen. And one of the cool things about cyanobacteria is that cyanobacteria are also autotrophs, which means that the cyanobacteria are fixing atmospheric carbon, not only atmospheric nitrogen, but atmospheric carbon. So it's the only organism in the planet that can fix both atmospheric carbon and nitrogen. Yeah? So this makes them very special organisms. Cyanobacteria, they are largely associated with fungi and structures that we call lichens. There are other types of free-living microbes that can be fixing atmospheric nitrogen, for example, Azobacter in the rhizosphere of grasslands. Yeah? And Azospirillum and Clostridium, they are also non-symbiotic, free-living bacteria that can be fixing atmospheric nitrogen in soil environments. Yeah? So ammonification is once the nitrogen is assimilated by plants, atmospheric nitrogen is fixed into ammonium, and this ammonium can be uptaken by plants and fixed into proteins and also into the bases that form the DNA and RNA. Now, whenever this organic matter, this plant debris or microbial biomass is dead and needs to be recycled, the nutrients need to be recycled, the process that removes that nitrogen and makes that nitrogen in the available form of ammonium again, it's the process that we call ammonification. The ammonification is the mineralization of organic nitrogen. Yeah? Mineralization of organic nitrogen. All the heterotrophic microbes, they're doing this. Yeah? This is not a very special and unique trait. All the heterotrophic community in soil, they are doing this. And the heterotrophic community in most soils, they account for the majority of the microbes. Over 90% of all the biomass they're heterotrophs. Yeah? So if you have that, you build up a high ammonium concentration in soils, you will have that you will cause ammonia volatilization. The reason being is that ammonium in soil solution is in equilibrium with ammonia in the atmosphere. The higher the concentration you will have of ammonium in soil, you will be inducing a higher rate of volatilization process. But that volatilization means that the ammonium is leaving behind one hydrogen, and that hydrogen needs to have a hydroxyl as a counter ion. And because of that, you have that this rate of volatilization is higher, is more pronounced when you have high pHs, alkaline conditions. And when you have that the moisture content in soils is decreased, then this also increases the rate of ammonium volatilization, because if you decrease the moisture content, you are concentrating the ammonium in the soil solution. And because the concentration of the ammonium is higher, you have a higher imbalance between the concentrations in the soil solution and in the atmosphere. This happens in very high rates when we apply ammonium-based fertilizers in soils. Yeah? But it's worth saying that ammonium in soils, they are not always in solution. Some of that ammonium can be associated with the solid phase and can be adsorbed. And by being adsorbed, it's protected from volatilization and also from leaching. So ammonium is less likely to be leached than nitrate, for example, in soils. So there are pros and cons to having ammonium as a source of nitrogen, and there are pros and cons of having nitrate. Actually, none of the inorganic forms of nitrogen are very stable in soil solution, in soil environments. Nitrogen is quickly lost. Yeah? As a fertilizer technology to prevent ammonium volatilization, you have some fertilizers contain urease inhibitors. That means that if you add urea to the soil, the inhibition of urease enzymes from the soil microbes will make that this urea is mineralized into ammonium in a slower pace. If urea is mineralized in a slower pace, it means that the concentration of ammonium will be not very high on the soil solution, and therefore the rates of ammonium volatilization will be lower, and you can have a more, let's say, slow delivery of this nitrogen for the plants. So this is a very common demonstrate that if you inhibit the microbial urease enzymes in soil, you have a higher efficiency of the nitrogen used from urea fertilizers. Now, when you add ammonium fertilizers to soils or nitrate fertilizers to soils, this will be immobilized on the microbial biomass. Whenever you have a carbon source in the soil, any organic matter that the microbes can use, the microbes will uptake this carbon, the heterotrophic community will degrade this carbon, and will need to take also a good amount of the nitrogen. So depending on how high the carbon to nitrogen ratio of the biomass that you add in the soil is, you will have that the speed of decomposition of this organic matter will change, and the availability of nitrogen on the soil solution will be changing accordingly. The reason being is, if you add a high carbon to nitrogen ratio residue, it will, as the microbes will start decomposing the carbon, they will uptake all the nitrogen from the environment. And because they uptake all the nitrogen will be deficient. So there will be lower formation of microbial biomass, a longer period of nitrogen depletion from the soil solution, immobilized on the microbial biomass. And therefore, this is highly problematic for plants. Plants will need the nitrogen, and if you don't have this nitrogen availability in soil solution, that will be detrimental for plant growth. So this nitrogen immobilization is a problem when you add residues with high carbon to nitrogen ratio to soil environments. While if you add very, very low carbon to nitrogen ratio residues, you have the opposite problem. The microbes will go too quick. It will be too easy for them, and you have an optimal carbon to nitrogen ratio. It will mean that its microbes will be growing too quickly and depleting the oxygen on the environments and creating anoxic conditions. So what we actually want is something like a 15:1 carbon to nitrogen ratio in the soils. And if you go beyond 20 and beyond 30, it means that you are having trouble and you are immobilizing the nitrogen in the microbial biomass. So this is something that you have to consider when you add organic matter to the soil. What is the carbon to nitrogen ratio of this organic matter? Will it cause quick growth of the microbes? Will it cause ammonium immobilization in the microbial biomass? Next step on the nitrogen cycle is nitrification. Yeah, nitrification is a very special process, metabolically speaking, because they're being performed by special type of bacteria that were first described by the father of soil microbiology, Sergei Vinogradsky, and the type of bacteria that are involved on this in this mechanism or chemolithotrophs. And chemolithotrophs are microbes that are using these inorganic molecules as a source of energy. And these microbes, they are usually, they can be important in more than one cycle. Some microbes on the sulfur cycle are very important on the nitrogen cycle and vice versa. And many of these microbes, they are autotrophs, means they are fixing also carbon from the atmosphere. The nitrification process, it's dominated by two bacteria. One is Nitrosomonas and the other one is Nitrobacter. Nitrosomonas will be oxidizing ammonium into nitrate, and Nitrobacter will be oxidizing nitrate into nitrate. And if you look at here, both of these reactions are rendering a positive yield of energy. So these microbes can use this reaction as a source of energy in the soil environment. So these are not heterotrophs, they're chemolithotrophs, autotrophic chemolithotrophs. The majority of the heterotrophs will be chemoorganotrophs, and these ones are using the source of energy coming from inorganic molecules, not organic molecules. The next thing it's important to say in this reaction is that this oxidation of the ammonium, it acidifies the soil environment. It leaves behind 4 hydrogen, and it means that, for example, that has implications for Oman. We have highly alkaline soils, and you should prefer to use ammonium-based fertilizers in Oman because ammonium-based fertilizers will be acidifying the soil environment. Now, if you are working on acid soils, then you have to preferably use ammonium, nitrate-based fertilizers, which will not be acidifying the soil environment. We have also nitrification inhibitors. Yeah, in some soils, the nitrate leaching and denitrification are the main forms in which nitrogen is being lost from the soils. So if you can avoid that the ammonium becomes nitrates and nitrates, you can have a higher efficiency of fertilizers. So the use of nitrification inhibitors is also used in those conditions. And high moisture content is one of the things that is an important condition to be considered. The amount of clays in the soils and the water regimes that you have on the soils are very important. This is a technology, obviously, to prevent nitrogen losses and increase nitrogen efficiency on these systems. So nitrogen leaching can be very important in the soils as a way of losing nitrogen. Nitrogen is not strongly associated with the solid phase, and it's mostly present in the soil solution. So every water that moves through the soil will carry the nitrate with it, and quickly this nitrate will reach the groundwater, the aquifer, and it becomes a health issue because high nitrate concentration causes blue baby syndrome. You know? And concentrations over 10 milligrams per liter, they're problematic on the water that humans are consuming. So we have to take care not to pollute our groundwaters, because groundwaters are one of the big sources of water for human consumption in the world. So another problem with the nitrogen losses to water systems is eutrophication. Eutrophication is if you enrich those water systems with enough nutrients, algae will grow, and while they grow, they create anoxic conditions on the water, which will kill the fish. And this is a big environmental problem, not only for lakes and reservoirs, but also here in the Gulf of Oman, there is a huge problem with the eutrophication in dead zones. And this is a problem not only here in the Gulf, but throughout the world. Eutrophication is a big problem. And this is being caused mainly because of the overuse of fertilizer in agriculture and the lack of efficiency of this fertilizer in agriculture. Now, denitrification in soil environments, this is an anaerobic process, and the microbes are not using it as a source of energy as in the nitrification process. Now these microbes are using it as a source of oxygen. They need the oxygen to be the final acceptor of electron. And in anoxic conditions, they don't have the oxygen, so they just use the oxygen bound to oxidize molecules like nitrates and nitrates. And in this process, it emits N2 back to the atmosphere. Now, this process is not entirely efficient, and some of this nitrogen is emitted as nitrous oxide. And this nitrous oxide, as we spoke before, are a problem for because it's a greenhouse gas. Yeah, it's a greenhouse gas, and it causes global warming. Now, the nitrate loss, the denitrification, is also it's a function of the concentration of nitrogen, but is also a function of the moisture condition. You have to remember that this process is anaerobic, and the more moisture you have more anoxic conditions, and this will be leading to more denitrification losses. The other factor that is affecting is pH, because in very acid conditions, this process is decreasing. Yeah, so you need neutral, approximately neutral to slightly alkaline conditions to have a quick denitrification process. So in acid soils, you will have very acid soils. You will have that this process will be less dominant as a nitrogen loss. But in highly alkaline soils with high moisture content, that will be a very important process. And temperature has a positive correlation with the denitrification, but only because temperature is affecting all biochemical reactions in soil and the microbial growth. So the higher the temperature, you will have also the higher denitrification happening on these soil systems. Another part of the nitrogen cycle that is only recently described is Anammox, anaerobic ammonium oxidation. This was only discovered in 1999. These are the reason that this was not known before is because these microbes are hard to culture. They're slow-growing microbes with generation time between 7 and 22 days. So it takes weeks sometimes for doubling the biomass of these microbes in cultural media. These are anaerobic microbes. But these microbes are highly important on the nitrogen cycle in water systems. And the reason they're highly important is because the enzymes they carry, they have very high affinity for the substrate ammonium and nitrate, which means that even in low concentrations of ammonium and nitrate, these microbes can be emitting atmospheric nitrogen into the environment because of the high affinity of its enzymes. So these enzymes are present on a structure in this bacteria. You remember that bacteria do not carry organelle because they're prokaryotes, but this seemingly organelle from this bacteria, it's called Anammoxosome. This is where the enzymes are, and they are responsible for this Anammox metabolism. Very interesting microbes. So the last part of the cycle is DNRA, dissimilatory nitrate reduction into ammonium, and this is also contributing for 15% of the total nitrate reduction rate. So before, every nitrate loss was attributed to denitrification, and only recently that we are separating how much of this denitrification is being due to DNRA and Anammox reactions. Yeah. Yeah. So that was for the microbial side of the cycle, but the microbial inputs on different branches of the nitrogen cycle have implications on nitrogen use from fertilizers and nitrogen nutrition of plants. So let's talk a little bit from the plant side, from the agronomic side. What are the implications of these processes on nitrogen nutrition of plants? So first of all, because nitrogen is so easily lost from soil systems, ammonium is volatilized, nitrate is leached and denitrified, and nitrate is also leached. The concentration of nitrogen and ammonium in soils is usually very low, and the majority of the nitrogen in the soil will be in the organic matter, either the living organic matter, the microbial biomass, or plant roots, or also in dead organic matter, stabilized or more or less labile in the soil. So the 95 to 98% of the total nitrogen in the soil will be usually in the form of organic nitrogen. The importance of this is the cycling of this organic nitrogen can provide a slow release and a steady supply of ammonium into the rhizosphere, into the soil solution, available nitrogen to plants. Whilst if you add only the chemical sources of nitrogen, these chemical sources will be quickly lost from the soil environment and will have limited impact on plant nutrition. time-wise, the effect will not last long. Whilst if you add, if you build up organic matter in the soil, you will have that you are contributing to the nitrogen nutrition of plants in a more steady and resilient way and more long-lasting way. So organic fertilizers are a good strategy of managing nitrogen in agroecosystems. But also inorganic nitrogen inputs are needed most of the time. Plants will take this inorganic nitrogen from the soil solution and will fix this organic nitrogen, inorganic nitrogen in organic molecules, and the plant residues can be cycled back into the soil organic matter. Yeah, the plants and also can directly receive nitrogen from the atmosphere through the nitrogen fixing microbes, mainly if you are if these plants are legumes with symbiotic association with rhizobium microbes. Yeah. So one of the main things that you have to take in consideration that when you are managing nitrogen in the crop is not how much urea you add, but you have to think about how you build organic matter in your soils. And one of the main strategies for building organic matter in the soils are green manure and cover crops. So what this means is you're going to add plants to your system in the off season, and these plants you will not sell them, they will not be your cash crops, but they are only there for building up organic matter and helping to condition the soils, protect the soils from erosion, and help nutrient cycling in these systems. The difference between green manure and cover crops is when you incorporate the plant residues in the soil, you call them green manure, classically done with lupine. In cover crops, you can have plants that will render high carbon to nitrogen ratio plant residues, but you will leave this material on the surface of the soil and not mix them with the soil as you would with green manures. But in either way, this is good soil protection mechanism, technology, and will build organic matter in the soil and will be a good conservation technology for these soil systems. This is some of the amounts that you can fix of nitrogen by using green manures that are also legumes. If you use the green manures or cover crops that are also legumes, you can have a lot of atmospheric nitrogen biological fixation in the soils. So, for example, sweet clover and crimson clover will be fixing more than 100 kilograms of nitrogen per hectare. This will be variable depending on how much is the biomass, how much is the amount of other nutrients you have on the system, and how efficient is the association of these plants with the rhizobium. But overall, it's a positive technology on the nitrogen cycle. You can supply nearly all the nitrogen that the crop needs only by having the cover crop. Now, if your cash crop, the crop that you are selling, is already a legume, you need to make sure that this legume contains the association with the proper rhizobium species. So in the way you do this is you can inoculate the seeds with the proper rhizobium species, and then you make sure that there will always be this rhizobium will be colonized in the roots of these plants. All right, the last option that you need to think about managing organic matter in agricultural systems and improving the nitrogen cycle in soil systems is the application of direct application of organic amendments. And different organic amendments will have different concentrations of nitrogen, but only by having high organic matter in the soil, that is already a good contribution and a good benefit for the soil overall. So independently of how much nitrogen you have on your organic amendment, this organic amendment will always be a good input for your soil, and it's worthwhile using it. If it's cheap enough that you can apply in high amounts, make sure you use and recommend those. Okay, that was all for the nitrogen cycle. Thanks everyone for joining. See you in the next lecture.